even in presence of organic matter. As well, the quaternary ammonia and aldehydes are high efficient in presence of organic matter. In the range of temperature, the only one which is active in low and high temperatures is the hydrogen peroxide and peracetic acid. I will explain later on about this. Also, biodegradable nature is only complied or fulfilled by hydrogen peroxide and peracetic acid. And these ones, at a diluted rate, they are non-corrosive. The only disinfectants that are corrosive are the chlorinated anyway. But complying with all these features, we only have one combination, one possible combination, which will be the mixture of hydrogen peroxide and peracetic acid. If we explain a little bit more about these chemical and physical parameters, you will see the differences, or why I say that hydrogen peroxide and peracetic acid is the only one which is effective, even though in those difficult conditions. If we study the effect of pH, for example, in contact with chlorine, I mean, when we use chlorine, for example, for water disinfection, pH of water is very important. Depending on the pH, chlorine formates or decomposes in hypochlorous acid or hypochlorite ion. If we have pH up to 7.8 approximately, the highest formation will be hypochlorous acid, which has a high biocidal effect, so we won't have problems. But if we have a pH over 7.8, in that case, the formation of hypochlorite ion is increasing quickly, and then we do not have a high biocidal effect. In fact, we have a very low biocidal effect. That is why when we have high pH over 7.8, as I said before, chlorine is not effective, because only hypochlorite ion is formed by being in contact, the chlorine, with the water. So we could state that chlorine is not effective. We also have some example about the temperature, how temperature affects the efficacy of different substances. In this chart, Taylor in 1999 was already comparing different disinfectants in clean and dirty conditions. They were comparing quats, combinations of quats and glutaraldehyde, chlorine dioxide, combination of peracetic acid and hydrogen peroxide, and just an acid detergent or sanitizer. In this case, at temperatures of 10 degrees, the only combination that was passing all the tests was peracetic acid together with hydrogen peroxide. In this case, we were comparing 0.5, 1, and 2% concentration against Pseudomonas aeruginosa and Escherichia coli. And the only product, the only substances that were passing in dirty and clean conditions, reducing a 5 log or greater in viable counts, was this possible combination. The other ones were failing in one or another case depending on the substance and depending on the test. But this was already tested in 1999 by Taylor. So we also have a direct effect of low temperature in different chemical substances. Of course, high temperature is also affecting to most of the chemical substances that can be available in the market for disinfection. Talking about the biodegradable nature of the disinfectants, we must state that hydrogen peroxide and peracetic acid are the only ones which are 100% biodegradable. Peracetic acid decomposes in acetic acid and hydrogen peroxide decomposes in water and oxygen. So these are all clean substances. There are no generation of subproducts, toxic subproducts that could affect nature, animals, or human beings. So already the European Commission, in its European Union Risk Assessment Report, declared the substance hydrogen peroxide as a 100% biodegradable substance. This is the time of degradation, degradation half-lives of hydrogen peroxide in different compartments. For example, in air, degradation will occur in 24 hours. In wastewater, 2 minutes. In surface water, 5 days, and in soil, 12 hours. Due to its rapid degradation as well as poor absorption, the European Union already declared the substance as a biodegradable substance. Taking into account all the details I gave you until now, we can conclude that according to the efficiency of disinfectants, we have two possibilities, which are aldehydes and hydrogen peroxide and peracetic acid. But as you see here, if we work with aldehydes, we need to label our product with a lot of symbols, danger symbols. I mean, we have toxic, harmful for the environment, corrosive, harmful, and irritant. So this is not a good substance because it is difficult to handle if we are keeping all these symbols in our labels. Hydrogen peroxide and peracetic acid have a high efficiency and some advantages. We just have to label with these symbols, which are harmful and corrosive. And as a conclusion, I could state that although aldehydes have a high efficiency, they have many disadvantages. They are toxic and suspected of carcinogen effects. They are difficult to manage and to handle. We need to be cautious in contact and inhalation, and we need a safety period. They are harmful to the environment. They are not biodegradable. They have low efficacy at temperatures under 12 degrees, and they have the possibility of creating resistance. However, talking about the hydrogen peroxide and peracetic acid, we could conclude that this is an environmentally friendly, totally environmentally friendly substance, which is not generating any toxic subproducts. And its efficacy is totally independent from pH, temperature, organic matter, any parameter that could affect other disinfectants. And finally, for its chemical oxidation, it is impossible to create resistance. So we could conclude that the best substance we can use nowadays for disinfection for a biocide is the combination of hydrogen peroxide and peracetic acid. This leads me to my third point, which is my third topic, which is the GOOX 100% biodegradable biosafety strategy. Why Grupo OX is working with these substances? We already know they are clean substances. They do not generate subproducts, toxic subproducts. They are easy to handle. But we need to know how we have to do things. So it's not just a substance. It is not just a biosafety program. Which is our key factor? Let's see. We work with hydrogen peroxide and peracetic acid as active ingredients, but we use the best quality active substances. The best quality active substances means that the hydrogen peroxide and peracetic acid we use has a lower quantity of impurities as raw materials. So you could find many types of hydrogen peroxide and peracetic acid, but these substances may have impurities. So we need to select the best raw materials in order to be able to stabilize correctly. So first thing is to buy or to use good quality raw materials. After we select those good quality raw materials, we need to stabilize. In hydrogen peroxide and peracetic acid, stabilization is essential. Hydrogen peroxide normally decomposes very quickly and normally has a shelf life of 6 months. So we really need to stabilize to have a wider spectrum of action and to have a greater time to use the product. And this stabilization must be done without adding heavy metals to the hydrogen peroxide. Normally you will find in the market hydrogen peroxide stabilized with silver nitrate, but silver nitrate is a heavy metal. That means that then we cannot state that this will be a 100% biodegradable product because it already has some metals in its composition. So we, as Grupo OX, were able to stabilize the hydrogen peroxide without adding any heavy metals to the composition. This is thanks to our R&D department, which is working in this stabilization course. All the OX products that are part of our biosafety programs have a different OX score. So each product has an OX score that makes the products more stable and more efficient against bacteria, fungi, virus, protozoa, parasites, whatever contamination we may have in water, surface, or environment. These OX scores are very important because they are masterful chemical formulas specifically designed for each product. And this graders or increases the efficiency of the products, even in different and hard conditions. Oxvirin is an ecological and effective disinfectant, especially designed to be used in facilities, tools, equipment, environment disinfection. It has been tested against different microbes, authorized by the UK DEFRA institution. And here you just have some tips of some efficiency we already tested against Salmonella. We have bactericidal efficacy against 5 strains of Salmonella after the first 5 minutes of contact at a very low dosage at 0.25%. We are also active against Campylobacter, inactivating Campylobacter by more than 5 log units in just 1 minute. We are also effective against virus, against avian influenza, inactivating in 1 minute at a dosage of 0.5%. Also, we are able to inactivate Coccidia oocysts at very low dosages. We are talking about inactivating 99.9% of oocysts in 60 minutes, less than 60 minutes at a dosage of a 5%, with a specific designed protocol for coccidia to prevent coccidia in broiler or piglets. Oxvirin is used in facilities as a broad spectrum disinfectant, but combined sometimes with another product, which is OX-S4, which is a dry powder specially designed for shock treatment disinfection in dry shape, not liquid, but dry. Here we also tested the hygiene, disinfection, and protection effect of OX-S4 against different microorganisms, total aerobic, fungi, and enterobacteria. We were working at a dosage of 100 grams per square meter. This is the initial result of total aerobic, fungi, and enterobacteria. After 1 week, we were reducing the counts very well, and after second week of treatment, we were reducing to half of the contamination we had in the beginning. In third week of treatment, we were more or less reducing also the contamination. We have a specific protocol depending on the contamination we have to work with this OX-4. But just as an example, I give you other results we obtained recently by using oxvidin and OX-4 in a specific work proceeding against Streptococcus suis. In this case, we were disinfecting facilities and equipment with oxvidin at a rate of 1%. Then we were also disinfecting the sows before birth with oxvidin at 0.5%. And finally, immersing the piglets with a mixture of oxvidin at 0.5% and OX-4. In this case, the results were very impressive as well, because we were seeing, in this test we had 14 sows, 183 piglets. In the control batch, we were treating 33 animals with antibiotics. However, in the treated batch, we were just treating 4 animals. In the control batch, we had 8 dead animals. In treated batch, we just had 1 dead animal. And the percentage of affected animals by Streptococcus suis was 22.4% in the control batch and 2.73 only in the test batch. This is going to be published in a few weeks in some technical magazines, because these results were very surprising after making all the tests. Also, OX-4 is meant to be used in a dry boot bath. This is last application we were developing, not to enter any humidity to some facilities by using a liquid foot bath. For example, in hatcheries, slaughterhouses, egg processing plants. They were asking us to use OX-4 in a dry boot bath to be able to discuss if it was possible to substitute liquid products by dry products. In this case, we were making some tests. Control of boot count was given us at the beginning 8 multiplied by 10 total coliforms, 36 E. coli, and 2 Pseudomonas aeruginosa. After putting the boots in the boot bath, we were also taking some samples, and then we reduced to 0 all contamination. And after 1 week of using OX-4, we were repeating the same results, keeping that efficiency along the time. Oxawa second generation is the product we developed for water treatment. This is also a product without chlorine in its composition, which is meant to be used in water treatment with very good results as well. Let's see some of them. In this case, we were publishing in the Sumac magazine, an Italian magazine, the effectiveness of Oxawa second generation for water treatment, the effect on fertility of dairy cattle and calf mortality. Before treatment, we were having a very high contamination of E. coli, microbes, total microbes, Pseudomonas, coliformi, fecal coliforms, and we didn't have Prototheca. But we had a huge count of these microorganisms before beginning the water treatment with Oxawa. After treating for 2 months, we were reducing drastically the contamination of water E. coli was given as just 4.41 counts. Total microbes, just 186. 5.6 total microbes have 22 degrees, and Pseudomonas 8.3. So if you compare these results with the results we had in the beginning before treating with Oxawa, after 2 months we were obtaining very, very great results. Of course, the product is meant to be used continuously to complete the disinfection of water and to assure we have zero contamination in the drinking points where the animals drink. But in this case, we were also comparing different data, treating and control. The number of animals we were treating were 608 cows. The days between the birth and the artificial insemination was 58.6 days with the treatment and 77.8 in the control batch. So we were reducing the days between artificial insemination and birth. Also, we were reducing the number of artificial insemination from 3.4 to 2.2. The period between birth and conception was also reduced from 148.6 to 104.5. Being pregnant after the third artificial insemination the percentage was raising very much as well, because we were raising from 73.4 to 84.3. And the best data was this one. The mortality of calves in percentage was reducing from 63 to 30, so from a 10.36% to a 4.9%. OX-Aqua Culture is a product mentioned to be used for disinfection in aquaculture. In this case, we also presented very good results in different tests we've been performing along the years, preventing diseases by using OX-Aqua Culture in shrimp and fish production, in the water treatment where the fish and shrimp is produced. For example, here I show you just one of our graphs. In this case, we are comparing reduction of, counts of bacteria without treatment, before treatment, and after treatment. As you can see, contamination, bacterial reduction was very, very quickly and very, very well observed. Treating with OX-Aqua Culture, with 25 ppm of OX-Aqua Culture, we were reducing very much the contamination of bacteria we had in water. Also, the survival of fish was very good, because we had a survival of 40% in the control batch, but at 40% with 25 ppm and a 40% of survival with 50 ppm. So there is a very, it is a very safe product. Indeed, although we are using 50 ppm of product, there is no risk to kill fish or to kill shrimp by using OX-Aqua Culture. So it is a very safe and very effective disinfectant for water in fish production. These disinfectants could be used in different business areas, in the animal health sector, in the plant health defense, in the public health, in the food industry. We are also working in invasive aquatic species and also in environmental hygiene services. The idea is to offer to all those business areas 3E global solutions: effective, ecological, and economically viable. And just to summarize what I was telling you today, and after my presentation, maybe you have some questions, we can assist you with any question you may have. Let me remind you some main conclusions I want to leave for you to remember. Well, First, before working, we need to make a previous technical audit. A previous technical audit with objective risk biosafety assessments are mandatory if we want to invest just the necessary resources in each facility. Secondly, it is a priority to adjust biosafety proceedings to each particular farm and implement customized solutions. Third, not all the products are similar, so premium products have to be selected for that intelligent biosafety management. But premium products do not mean expensive products. They do not have to be the most expensive products in the market. So we really need to select what we really need depending on that previous audit we have to perform. And finally, the intelligent GoWorks biosafety strategy helps to increase shelf life of materials and equipment, to improve hygiene and health status, reducing disease incidence, to reduce spending on medical treatments, and finally to improve soil technical indexes, improving therefore productivity of farms. So biosafety means prevention, biosafety means money investment and money saving. Well, thank you very much for your assistance today. Any questions you may have, I will be delighted to assist you. Well, in any case, we are located in Pavilion 105. Our booth is the L44. So if you need any further assistance, I will be pleased to help you there and to respond to any questions you may have. Thank you very much for your assistance, kindness, and time today. In that graph I presented, there was one part which was we were working in water treatment, but also in the protocol we were working in some disinfection in the environment of the farm. So the complete technical article is already published in this.
Oh, well, I don't have any doubt on the technical article. Yes. My question is very simple and straight, that can we make a claim that if you use this product, fertility will be improved that much?
Well, it's part of a protocol, okay? But we saw that this was made in a very high qualified farm, a cattle farm, and we saw if we were working with our protocol, we improved with that data. So working on biosafety means we are having very much better productive results. In this case, we were working in disinfection, and we were having those results. So we must state that working on biosafety and working on a specific strategy in biosafety, we can improve in a reduction of calf mortality for sure, yes.
If that is because of the biosafety problems, if it's some other problem, maybe the claim will be too big.
Of course. I mean, this is part of a working proceeding, which also needs all the parameters to be controlled. I mean, yeah, biosafety is part.
Once you state this thing, it's a very big and wild claim that, I mean, just using disinfectant, you improve the fertility.
No,
there might be several other things as well. Okay.
No, it's not just by using a disinfectant. You need to continue with some proceedings, yes. Any other questions? Well, thank you very much.
There are a lot of other factors obviously involved in egg quality as well. And I'll touch on some of the common things you'll see in the field and help you a little bit with diagnostics on eggshell quality problems and overall egg quality problems. Egg quality really is an integrated process. The egg starts on the farm, and we've got to get it delivered to the customer's table in the form in which they expect it. And nature actually has provided the egg as this perfect encapsulation of food. And it's very nutritious. It's also delicious. which is why we get great customer demand for it. And it's essentially our work in the industry to preserve getting that food from the farm to the table. Some of the things you need to think about in the concept area, markets. There are big market preferences. Here in Southeast Asia, brown eggs dominate with the exception of the Philippines. In North America, white eggs are common. In Europe and in South America, many markets actually have browns and whites together in the same market. So it's very much a market-dependent thing what the color preference will be. The size of the egg is another market trait. There are big egg markets in the world, and there are markets that are looking for eggs in a more medium-sized range. And actually, we've seen a lot of tendency for the larger egg markets to drift towards requiring an egg somewhere between a 55 gram to a 60 or 65 gram range. And if you looked at historic egg production and egg weight ranges, only 20 years ago the birds would come into lay with a 40 gram egg weight and they would go out of lay with a 70 gram egg weight. That meant that 15 grams were outside the consumer accepted range back then, and so they were undergraded and they were sold at a greatly reduced price. Some of the limitations to maintaining egg quality are actually the production chain, things that happen to the egg as it gets from the farm to the table. Other limitations can be market size and the distribution system used to deliver that egg. You also need to remember eggs are consumed in two ways. One is the shell egg delivered to the customer. The other is eggs that are presented to the customer already in products such as bakery products where it's been processed. And there are impacts from companies and agencies. We have to make sure that the egg is healthy and free of bacterial contamination. And also we have government agencies usually classifying eggs by size and other characteristics that would be considered quality. When you look at egg quality, we'll tend to always break things down into the shell, the exterior. Here we're looking at things like thickness, but also form and texture. People expect eggs to have a smooth form to them. They're looking for freedom from some of these superficial defects that can occur on the eggshell surface. We also, of course, place great emphasis on resistance to breakage, but color is very heavily emphasized and even sheen on the egg. Also of importance, though, are the antimicrobial factors. There are proteins both in an eggshell as well as in egg white that have antimicrobial factors. In the eggshell, some of those proteins would be things like ovocalixins, which people don't commonly think about. And of course, the physical structure of the eggshell can also determine how well it's protecting from microbial contamination. Interior qualities, again, the microbiological characteristics are important, but why do people buy eggs? They buy them for the taste, but also how versatile they are in cooking. But also smell can be an important characteristic. In the past, we used to struggle with brown eggs. You had to be careful how you fed brown birds in the past because you could get this fishy taint smell. That actually is something breeders have been able to remove from the populations. Yolk color and yolk texture are also extremely important. Most of the organoleptic, in other words, the taste and the smell and the texture of an egg, actually come from yolk characteristics. Egg white tends to be a rather bland, odorless, with a difficult texture to work with unless you're using it for foaming properties in baking. Albumin height is typically measured as a Haw unit, and a Haw unit is just an albumin height corrected for the weight of the egg. So as eggs get bigger, their yolks get bigger, so of course it pulls the egg white up. So that's why the Haw unit is used as a standardized measure of the egg white quality. Yolk size has become much more important in recent years, and also the vitelline membrane, or that membrane around the yolk, because industrial eggs are often separated at the point where they're broken, and making sure you don't get yolk contaminating the white, and also you want to get as high a yield of yolk as possible, because yolk usually has 2.5 to 3 times the value of egg white. So you see selection for yolk size going on today to give higher value to the egg. That results in a better total egg solids. And remember, most of the nutritional value in an egg actually is in the egg yolk. The other thing we're looking for on interior qualities are presence of any inclusions, the so-called meat spots and blood spots. While I'm a geneticist and I think about the world only in terms of genetics, I do realize it's important those interactions that occur from a nutritional standpoint, from a health standpoint, management and environment. But I will focus a fair bit on the genetics of egg quality.